Hi, welcome to my YouTube channel. Today I'm going to discuss 25 memory-based ASRB NET prelims as well as Senior Technical Officers 2021 exams question paper on plant pathology. Let us start with the first question. The question is, the complex disease cycle of cedar apple rust alternating between two plants, that is host plant, was first delineated by? The correct answer is Anders, Sadiu, Orsted. Cedar apple rust, which is caused by Gymnosporidium juniperi virginiani, is a fungal disease that depends on two different species to spread and develop. It spans a portion of its 2-year life cycle on eastern red cedar apple, that is Juniperus virginiani. The scientist Anders Sadiu Orsted, he was born on 21st June of 1860 and died on 3rd September 1872. He was a Danish botanist, mycologist, zoologist as well as marine biologist. In his early years, he worked on Danish and Arctic nematode and marine algae in Orsted, commonly known as English as the Sound, and is a strait which form the Danish-Swedish border separating Jylland, that is Denmark, and Scania of Sweden. He worked on juniper pear rust, as this fungi is heteroecious in nature, as it switches its host between the primary host, that is teleal stage Juniperus savini, and secondary host, that is acial stage Pyrus communis of the pear. The orchid genus Ostridiella rake handbag is named after him for his excellence of work. The cedar apple rust, it produces different type of symptom. First one is gall on cedar trees. Here the disease begin on the cedar or the juniper trees. Small, round, gelatinous and light brown to orange-brown galls or the swelling form on the branches of the cedar trees. These galls may have a small spore-producing structure called talia on their surface. Another symptom, that is yellow spot on the apple leaves. On apple trees, the first symptoms on leaves appear as a small yellow to yellow orange spot on the lesion. These spots may initially be hard to notice. Another symptom produced due to this disease is bright orange circular spot. Here, as the disease progresses, the yellow spots on the apple leaves develop distinctive bright orange circular spots with reddish border. These spots often have a raised appearance and are more visible. Another important symptom, that is lesion on the fruits. Cedar apple rust can also affect the fruits of the apple tree. On infected fruit, small circular rusty orange lesions or spot may develop. These lesions can cause fruit deformities and reduce fruit quality. Followed by another symptom, that is premature leaf drop. Because of this disease, at severe stage, it may cause to premature leaf drop on the apple trees, which can weaken the tree and reduce fruit production. Followed by the impact on cedar trees because of this disease, that is cedar apple rust. Here, while the impact on the apple tree is more noticeable, cedar apple rust can also affect cedar or the juniper trees by causing the formation of gall and weakening the branches. Followed by another question, triple gene block protein guided cell to cell movement is present in, and the correct answer is Pox virus. As we know, Pox viruses are a group of single-stranded RNA viruses. They belong to the family alpha flexi-viridity. The potex virus genome consists of a single-stranded RNA molecule that codes for several proteins essential for virus replication as well as spread. So, one of the key features of the potex virus is that the triple gene block, that is TZB protein, which plays a crucial role in cell-to-cell movement of the virus within the plant. And the TZB protein are expressed from 3 separate overlapping open reading frames, that is ORFs, in the viral genome. Each TZB protein has its specific function and cooperates it in facilitating the movement of virus through plant cell. And try to remember, the 3 TZB protein in potex viruses are typically named as TZB1, TZB2 and TZB3. Out of these 3, the TZB1 protein is involved in RNA silencing suppression. And RNA silencing suppression is a natural defense mechanism in plant that target and degrade viral RNA to prevent the virus spread. And TZB1 always help the virus counteract this defense by interfering with the plant's RNA silencing machinery, allowing the virus to evade detection and continue its infection. Secondly, the TZB2 protein is always responsible for enhancing the efficiency of cell-to-cell movement. It enables the transport of the viral RNA from one plant cell to the adjacent plant cell through plasmodesmata, then the channel that connect plant cell. The TZB2 plays an important role in enlarging 3 channels, which facilitates the virus movement between cells and enables systemic infection within the plant. And third protein, that is TZB3, is always involved in the assembly of the virus particle and essential for their intercellular movement. It contributes to the formation of the viral replication complexes, which always aid in the replication and the packaging the viral RNA, preparing it for further infection. The coordinated action of the triple gene block protein is critical for successful virus infection and spread within the plant. And understanding the function of these 3 proteins help in devising strategies to control the spread of potex viruses and other related plant viruses. Third question is, rosetting of leaves is due to the deficiency of? This is a very simple question and the correct answer is zinc. Generally, the zinc deficiency occurs in calcareous soil, and this deficiency lead to Khaira disease of rice, white bud of maize, little leaf of cotton, myrtle leaf of citrus, rosetting formation in majority of the agricultural crops, and parakeratosis in animal as well as in human being. Next question is, the symptom of aluminum toxicity in plant is? There are 4 options. Out of this, the correct answer is induced root growth. The rosetting always occurs mainly due to the zinc deficiency as explained in the previous slide. And yellowing generally occur mainly due to the deficiency of the nitrogen, but in some crop, yellowing also appears due to the deficiency of other element like iron, that causes iron chlorosis, or yellowing in groundnut, and yellowing of tea leaves due to the sulfur. Next question is on yellowing of young leaves is due to the deficiency of? Out of these 3 options, the correct answer is sulfur deficiency. Regarding zinc deficiency symptom, which has already been explained just few minutes back. Besides that, boron is another important element, and because of which lot of deficiency symptoms occur in agricultural crops. The common symptoms are fruit cracking of tomato and pomegranate, browning of cauliflower, top sickness of tobacco, heart foot rot of citrus, hand and cheek disease of. This is another question. Dash is a new class of argine included that acts majorly against plant viruses. There are 4 options. First is eukaryotic translation initiation factor, that is EIF4E/4G, then recessive gene, that is SDM1, LRR NBSCC, and last option is LRR NBS-TIR. Out of all these 4, the correct answer is eukaryotic translation initiation factor, that is option A. Next question is on gene for gene hypothesis in 1946 is given by, the correct answer is H. Floor, who proposed gene for gene hypothesis in linseed or the flax rust caused by Melapsora lini. It is a widely accepted model explaining the molecular basis of interactions between plants and their pathogens, particularly in the context of host resistance and pathogen virulence. According to the gene for gene hypothesis, plant possesses a specific gene, that is R gene, that encodes resistance protein, followed by pathogens such as bacteria, fungi, viruses possesses corresponding gene, that is called your avirulent gene or aviar genes, that encode avirulence protein. And thirdly, I want to say the resistance of a plant to a particular pathogen is determined by the presence of a specific R gene in the plant and a corresponding aviar gene in the pathogen. Most importantly, the key points of the gene for gene hypothesis proposed by Floor is that when a plant with a specific R gene encounters a pathogen that possesses the corresponding aviar genes, a molecular recognition event occurs between the product of these genes. And this recognition leads to a defense response in the plant, activating resistance mechanism that can prevent the pathogen from successfully infecting the plant. Interestingly, on the other hand, if the pathogen lacks the avirulent gene, that is aviar genes, corresponding to the plant's R gene, there is no molecular recognition and the plant remains susceptible to that pathogen. Another important point is that the gene for gene hypothesis explains why some plants are resistant to certain pathogens while others are susceptible, and it provides a molecular understanding of plant as well as pathogens interaction. Next question is on a well-characterized damage associated molecular pattern, that is DAM. Out of these 4 options, the correct answer is cysteamine. Next question is on a single cell conidium is called as ameroconidium. Besides that, as we know, there are other different type of conidium. Say we can cite the example of didmoconidium, means 2-cell conidium. That means 2-cell conidium are known as didmoconidium. Besides that, we can take the example of phragmoconidium. Here the conidium having 2 or more transverse septum. This type of conidium are known as phragmoconidium. Next question is the mode of action of strobilurin fungicide is inhibition of electron transport in ETS, that is QOI. The mode of action of strobilurin fungicide is totally based on their interaction with the fungal respiration process, which is specifically by inhibiting the mitochondrial electron transport chain. And we have to remember that the key mode of action of strobilurin fungicide involves binding to the cytochrome BC1 complex, that is complex 3, in the mitochondrial electron transport chain of susceptible fungi. And this interaction always disturbs the flow of electron through respiratory chain, leading to a reduction in the energy production, that is ATP synthesis, in the fungal cell. Because of this, the growth and development of the pathogenic fungi are always hindered. Ultimately, it leads to their death. The step-by-step explanation of mode of action of strobilurin fungicides are: number 1, fungal respiration. That means fungi, like other eukaryotic organisms, generate energy through cellular respiration, which takes place in the mitochondria. And the respiratory chain consists of several protein complexes, that is 1 to 4, and that transfer electron through a series of redox reactions, ultimately leading to the production of ATP and the cell's energy currency. Secondly, binding to the cytochrome BC1 complex. I mean to say, strobilurin fungicide specifically target and bind to the cytochrome BC1 complex, that is complex 3 of the mitochondrial respiration chain in the fungal cell. In the third point, I want to say, they inhibit the electron transfer by binding to the cytochrome BC1 complexes. Strobilurin fungicide always disrupt the flow of electron through the respiratory chain, and this interferes with the transfer of electrons to the complex 4, that is cytochrome C oxidase, and which is the final step of the electron transport chain. In the fourth point, that is ATP depletion. So as we know, as the electron transfer is hindered due to the action of strobilurin, the normal flow of the proton across the mitochondrial inner membrane is always disturbed, and consequently there is a reduced proton motive force that lead to the decreased ATP synthesis. And the lack of ATP severely impairs the essential cellular processes, ultimately leading to the death of fungal pathogen. Next question is, fungicides that are involved in the activation of SAR, that is systemic acquired resistance. The correct answer is benzothiazole or BTH. So before answering this question, we should understand some of the point, like say what is the role of some of the other material like salicylic acid and its analog. So we have to remember that salicylic acid is a natural plant hormone that always involved in the regulation of systemic acquired resistance. And the chemical analog of salicylic acid, so we can take the example of acetbenzolar S-methyl, that is ASM, and which is generally traded as Actigard. It can be used as a fungicide to induce SAR in the plant, leading to the enhanced resistance against various pathogens of agricultural crops. On the other hand, BTH, that is benzothiazole, which is traded as Bion, is another chemical inducer of the plant defense responses as well as systemic acquired resistance of the SAR. When applied to the plant, this BTH, it always activates the defense related gene and enhances the plant's ability to resist the pathogen attack. And then another product, that is herpin protein, which are bacterial derived protein that can trigger plant defense responses including SAR. They are sometimes used as a foliar spray to induce systemic resistance in plant. Next question is, amphimovile fungicide is, the correct example is strobilurin group fungicide. They are always amphimobile in nature. Another question is, the bitter pit of apple is due to, the correct answer is calcium deficiency. Beside that, due to calcium deficiency, some other symptom may develop, as for example, blossom and rot of tomato, tip hooking, death of terminal bud, and many more. The 14th question is on the genus that belong to Gemini virus, which is transmitted by aphid. Out of the 4 options, the correct answer is D, that is Capulovirus. Let us try to understand about other options. Here, I want to share some of the example of baculovirus that are transmitted by insect. Number 1 is Autographa Californica nuclear polyhydrosis virus, that is ACNPV, which is transmitted by moth Trichoplusia knee, followed by Bombyx mori nuclear polyhydrosis virus, that is BMNPV, which is transmitted by silkworm moth Bombyx mori. Then Oryzophilus mercator nucleohydrosis viruses OMV, which is transmitted by Khapra beetle Trogoderma granarium. Next, cutovirus. Cutoviruses are not transmitted by insect. So they are a group of virus that infect plant, and they are spread by contact with infected plant material. There are no known cases of cutoviruses being transmitted to humans or animals by any insect. But the name cutovirus comes from the Latin word cutis, which means skin. And cutoviruses are named because they often cause a symptom on the leaves of the plant that resemble cut or lesions. Some common cutoviruses include cucumber mosaic viruses, tobacco mosaic viruses, then tomato spotted wilt viruses, etc. Then followed by, say, option C, grabuloviruses. These are always transmitted by tree hoppers. And this grabuloviruses is a genus of Gemini viridae family. And regarding the option 4, there is a correct answer. The Capulovirus is a genus of Gemini viridae family of the viruses, and aphid always transmit this genus to. Next question is on which of the following is a embryo-borne viruses. Out of the 4 options, the correct answer is BCMV. On the other hand, TMV is always transmitted through mechanical inoculation, and then ICMV, that is internal cassava mosaic virus, is transmitted in a persistent manner by the whitefly Babesia tabaki, and the infected planting material always serve as source of infection. Then CMV is transmitted through insect. BCMV, that is bean mosaic virus, is transmitted by pollen as well as aphid. This is an interesting question. That is non-persistent virus that follow capsid only strategy for its insect transmission. There are, say, 3 options: CAMV, CMV, and rice dwarf. Out of these 3 options, the correct answer is CMV. Next question is on internationally reputed national collection of plant pathogenic bacteria is under the administration of. The correct answer is United Kingdom, that is option C, UK. Next question is on the post-graduation course of plant pathology was first introduced in. Out of the 4 options, the correct answer is B, that is Cornell University. And the post-graduation of the plant pathology for the first time introduced in our country, that is India, in the University of Agricultural Science, Bangalore, formerly known as Mysore Agricultural University. The temperature used for glassware sterilization in hot air oven? This is another question, and the correct answer is 160 to 180 degrees centigrade. So beside that, we have to remember what should be the exposure time. That is, at 160 to 180 degrees centigrade, the exposure time is near about 2 hours. On the other hand, at 60 degrees centigrade, it should be exposed for 4 to 5 hours. And at 240 degrees centigrade, we should expose it for 1 hour. Next question is on average number of protein coding genes present in the typical fungal genome is, the correct answer is 10,000 genes. Next question is on average number of total genes present in 5 to 6 MB plant pathogenic bacterial genome is, the correct answer is 6,000 genes. So I can cite some example like Pseudomonas syringae, which is a common plant pathogenic bacterium. It has a genome size of 5 to 6 MB and contains roughly 4,000 to 6,000 genes. On the other hand, Xanthomonas campestris, this is another plant pathogen. It has a genome size of approximately 5 MB, contains around 4,000 to 5,000 genes. On the other hand, another example, that is Ralstonia solanacearum, which is a common soil-borne bacterium causing bacterial wilt in many important agricultural crops. It has a genome size of about 5 to 6 MB and it contains around 3,000 to 4,000 genes. The next question is on dash is the range of wavelength for induction of conidiation in the general fungi, and the correct answer is 350 to 500 nanometer. And in general, the UV light wavelength is beneficial for induction and germination of conidia of Botrytis cinerea. Next question is on the race and the disease of Ralstonia solanacearum that is not reported from India, and the correct answer is mulberry race, that is race 5. Generally, in our country, 4 types of races of Ralstonia solanacearum are present, but race 5 is not present and reported in India. Next question is on disease of apple that is not yet reported from India. The correct answer is fire blight of apple. Beside that, we have to remember European canker, which is caused by Neonectaria ditysemia, and apple mosaic virus, that is APME, are not yet documented or reported from India. Next question is on toxoflavin is generally produced by, the correct answer is Burkholderia gladioli. So let us understand what is toxoflavin. Toxoflavin is a yellow toxic compound that is produced by certain strain of bacterium like Burkholderia glumi, and which was formerly known as Pseudomonas glumi. And the toxin, that is toxoflavin, is involved in the breakdown of the plant tissues and the colonization of the plant by the bacterium. In rice plant, the Burkholderia glumi infect the panicle, that is the reproductive structures that hold the rice grains, and causes the panicle to turn totally brown in color and resulting in panicle blight. And this disease can lead to the significant yield losses in rice production. Thank you, my friend, for watching this video. For next part, please keep in touch. Thank you.